Hi, everyone. Thank you so much for coming to our first AI Institute seminar series of the quarter. I'm really happy to introduce Dr. Abhe Rajagopal, who's a senior AI research scientist at the Allen Institute. He received his PhD in electrical and computer engineering from UC Santa Barbara, and before this, he held research scientist positions at Altos Labs and Tyon Research. He's also been a postdoc fellow at UC San Francisco in the Department of Radiology and Biomedical Imaging. And his research is really focused on building multimodal machine learning and computational imaging techniques to improve our understanding of human biology and pathology. So today he'll be talking about his research, and I guess I'll take it over to you.
Great. Thanks for the introduction. Could everyone hear me online? Should I mute my speaker? Are there speakers in the room? We have questions or? Okay, cool. Okay, thanks for the kind introduction. Yeah, today my talk is opening the black box of biology, of course, like everyone's talks with AI. But really, it's kind of the culmination of the last maybe 7 years of work, trying to piece together different aspects of using AI to try to understand biology. So recently, in the last year or two, I've been at the Allen Institute, which is right across the small pond that we have, but also have an adjunct professorship at UCSF, where I can continue my collaborations with the medical school. So I thought I'd start with how I think about health and medicine and disease. Really, to understand biology, think of it as a multimodality inverse problem. We know how we are and how we behave, but we want to make measurements to look inside of us because we can't see them with our naked eye. So what are these measurements? Well, you might be familiar with MRI or PET, CT, or ultrasound, something that you can observe by putting sensors on the outside of your body. But of course, we could also open up your body and excise tissue and look at it under a microscope. In tissue microscopy, we can stain it and look at it with fluorescence markers and things like that. We can go one level deeper. We can look at what is in our cells. So RNA-seq or attack-seq. RNA-seq, for example, is a way to measure what your cells are expressing in terms of genes, specifically in terms of the proteins that the RNAs that are encoding, and we use that to back out, okay, what is the expression of various genes that are in our cells? We can go one level deeper than that. We could ask, what is the DNA that's in our cells? And we can sequence our DNAs, ATGC. We can get the entire sequence and we can identify mutations and what's different across between us and what's similar, or across different species. We can go one level deeper than that. We can look at the actual structure of the DNA or the proteins that are encoded by the DNA and look at things like chirality. For example, some chiralities of some molecules can cause disease, or Alzheimer's as an example, and others, maybe it's just fine. And the way we would get at that is by using measurements, right? And so there's different types of microscopes and measurement techniques. Of course, we can measure these things. But in health and medicine, it's not enough to get that measurement to be able to measure the structure, even the atomic structure of some of these items. What we're really interested in is something that's a little bit more hidden than that. What do those measurements tell us about health? So as an example, you might be able to make an MRI image of the body, a whole body, or a particular organ, but we don't know, that doesn't by itself tell us which parts are working or which parts are dysfunctional. As an example, we might look for biomarkers of healthy versus disease, but what's really interesting now is looking at biomarkers of disease survivors. What are the mechanisms that actually allowed those patients to respond to therapy or cure themselves through using their immune system? And so a way to put this together is kind of looking at what are the causal factors in biology, and importantly, if we can use all the set of measurements to learn a health state for every person or every tissue or every cell, that is able to predict or forecast how that object will respond to intervention or how it'll progress in time. So this is kind of maybe the challenge in medicine, is coming up with all of this part lists and how they're interconnected, just really using measurements that we can make. So you might be familiar with the idea of inverse problems, looking at integral equations and trying to solve the values on a domain given measurements on the boundary. And especially in these sort of computational imaging scenarios where we know the physics of the forward operator, we can write it down, let's say, as an example, as a optimization problem, where maybe we have a data consistency term. Here, let me turn it on. We have a data consistency term, and then you have some regularization. And especially when this functional is convex, we can prove guarantees. We can have guarantees on how well it's converging. And of course, it's not immune. Deep learning can be used to learn this regularization with actually strong guarantees. And actually, I gave a version of this talk last year in the ME department, kind of looking at some of those different aspects in using deep learning for regularization. But the topic of today's talk, I thought I'd try to focus on, let me go here. I thought I'd focus on what we do when the physics is not known. And in that case, we tend to turn to machine learning or some sort of data science to figure out what the connection is between these inputs and outputs, or the measurements and what's hidden. But it's really difficult because not only are measurements high dimensional, we don't have really good validation of what are the parts in the human body. What are the parts in the cell? And what do they do? We don't exactly know that. So how do we solve that problem? And importantly, just because we're able to make a deep learning algorithm that's able to take measurements and, let's say, predict these inner parts of our body or these inner mechanisms, what does that tell us? Is it the fact that it's a predictive model? What does it tell us about how these objects are working together? So just to provide a little bit of context before I jump in, most of the talk I want to focus on how we build up a theory in the same way that we approach or look at physical inverse problems, and we have a theory for understanding when it's consistent, typically because it's pretty well characterized in the forward operator. How do we build up that theory of what the forward operator is for, or the inverse operator, let's say, for these type of medical biological questions? And to provide some context, though, before I start, I have this grant right now with UCSF and a few others where we're looking at multimodality data, spatial transcriptomics, which refers to looking at the gene expression data in individual cells, but in a spatial configuration, so meaning that you can align it with tissue microscopy. And so this is like two complementary types of information, like what's the structure or morphology of a cell or a tissue, together with what genes is it expressing. So it's a way to connect essentially structure to function. So that's, this is the focus of the grant. And, my portion of the grant is to use existing foundation models and look inside and figure out what actually connects structure and function. And so I'll talk a little bit more about this a little bit later, but I wanted to provide a little context before I jumped into how we think about this theory. So to start, I thought I would start with something that we can all sort of set the basis for, right? Or foundation for, and that's really approximation theory 101, right? Which is basis for neural networks and polynomials and curve fitting and all this other stuff. And one of the classic results there is that when we're in high dimensions, we need an exponential number of parameters to represent that function. And as a result, we think that we need an exponential amount of data to fit those parameters. But what's really surprising about this, and so you can plug in a few numbers and see how that curve scales, and of course the constant's missing, but you can see how the curve scales. But I guess what's surprising is that deep learning really works, and it has way fewer parameters with respect to the dimension that we're looking at. So images, even relatively small image chips, are really high dimensional mathematically, right? But our deep networks have way fewer parameters, and importantly, we don't have that much data to train it. So why do they work? And so the idea is that it must be exploiting some sort of structure, and the question is what structure and how? Well, maybe 10 years ago now, Moscar and Poggio proved that if you have the real compositional structure of the function that you're trying to approximate, what they proved is that if that compositional structure is deep and in form of like a deep architecture, or in the sense of multiple layers and functions feeding into each other, then they prove that you can escape the curse of dimensionality. And so the way that works is, or the kind of the crux of that proof or that theorem is that each of the interior nodes of this directed acyclic graph representing the function, the true function, operates on a lower dimensional space. And because of that, through function composition, they can show that you can actually escape this curse in some sense. And so basically the number of parameters you require grows, where d grows with d in the exponential, but based on the nodal degree of the function. So this is a really simple example, of, let's say a 2-layer network. And so as an example, you could pick the max function. What's the max of 4 items? That's a 4-dimensional problem where there's 4 different inputs, x1 through x4. But at the first stage, all you need to do is take the max of 2 items. And then you can apply that exact same function multiple times, and you can get the result for max 4. And so one of the really interesting things about this is that part of their proof was that you have this additive composition property. And importantly, by looking at derivatives using chain rule through the network, they showed that you can bound the overall gradient of the input of the entire function using the gradients essentially, or the Sobolev norms of the interior functions. So they kind of proved an error bound, a generalization bound, which depends on the infinity norm of the gradient of the function, or the Sobolev norm. So less than that, maybe 7 years ago, when I published my thesis, I was able to extend that a little bit. The problem with the infinity norm is it's hard to compute. It's not amenable to stochastic gradient descent. You can't use it as a loss function and so on. So I did some analysis, and honestly, pretty tame stuff. But basically what I showed is that, and this is a more complicated version of the result that corresponds to the same case that, the setting that they're using. But I really like this form better because it kind of highlights the terms. But basically the point is that the generalization error depends on 2 things, and I'll give you a sketch of the proof in just a second. But the first term depends on the training error of the Sobolev norm at the training points that you have. And then the second term depends on how dense your data is. And this really matches, I guess, the intuition of their original theorems. So just to give an example, and I don't want to belabor this point too much for the sake of time, but happy to come back to it after. As well. But basically the idea is that we're just going to use Cauchy-Schwarz to bound each of these terms. And then, for step 2 and step 3, for E1 and E3, use mean value theorem. But the key takeaway I want you to get before we move on is that deep networks use a composition of really simple low dimensional nodes to encode really complicated functions. So that's point 1, and that's from Oscar Poggio almost directly. The second part of their theorem was that local node properties, in particular the Sobolev norm, or the data density and the gradient of each of the individual nodes, affects the overall generalization ability of the model. And the third insight, which I think comes more directly from, I can see it a little bit more directly in the theorem that we proved, or the error bound that we proved, is that if you have smooth nodes and dense data, you have a lot of data on those nodes where this training distance, the distance to the density of the data is high, you get good generalization properties. So I want to give you a really simple example, something you can visualize with that same function max d, which is max in d dimensions. So let's say in 2 dimensions, if I train just with an MSE error, which is mean square error as the loss function, and you give it a limited amount of data, and you visualize and you plot for many other input-output pairs, what's the value, you find that the function that a neural network will learn, if you let it be free, can be quite rough. You need enough data to get it smooth. But explicitly adding the Sobolev penalty, which actually doesn't depend on adding more data points, all it looks at is the smoothness of individual nodes, and you're trying to minimize that with respect to the data points you already do have, you can essentially stabilize the training. So this works in 1-layer networks. Of course, it's pretty simple, but it also works in deeper networks. And in particular, the problem gets a lot worse as you get deeper. If you don't have data density, the problem gets significantly worse. Where, as an example, this is in the first layer, and there's 4 different input nodes. And you could see that the data is much more rough compared to using this sort of Sobolev norm penalty or controlling the gradient. And of course, it works in higher dimensions too. But what we find is that empirically, and there's not a theorem for this because it's using a finite amount of data, but empirically, we seem to avoid the curse of dimensionality with respect to how many data points we need as well. Meaning that maybe because there's fewer parameters to estimate, maybe you need less data as well to estimate them. And so that empirically, that's what appears to be the case. So the takeaways are actually the same here, right? We're using a deep network to get composition. We're looking at the local nodal properties, specifically the Sobolev norm and the data density. And we're seeing that when you have both of these properties being true, they lead to good generalization. But I started this whole thing with, we don't know the true function of that we're trying to approximate. We don't know what the forward operator is for the inverse operator is for a lot of medical or biological inverse problems. So this is where we need to make a little leap of faith. But the first leap of faith is that if you have a deep learning algorithm that really does work, it really can predict for every conceivable pair, input-output pair, it's well trained and it can predict the output given the input, maybe we can believe that the model architecture contains a subset or it's able to encapsulate the actual graph of the true function. And so we're going to try to look at that function as a surrogate for having the real graph of the target function. The second idea is more of a hypothesis, and the idea is that if foundation models are really learning biology, then if we go look at their nodal functions, we should be able to extract out that biology or be able to predict real physical, biological, chemical relationships between the entities that they're modeling. Like in the same way that we were looking at the nodes of the max function, we should be able to go look at it and say, what is that node computing and how does it depend on the inputs and what is the smoothness of it? Where is the data density? And specifically, what is it modeling? So, okay, the question is, so the idea is that, well, especially the second point, this is actually what people do already, right? So if we look at the anatomy of like a foundation model, and let's say specifically a biofoundation model, this is the recipe, right? The recipe is get a really big data set, build a model architecture. You pick your favorite one, a CNN, a transformer, whatever it is, and train that network with enough regularization and supervision that it's able to learn the structure, hypothetically. For example, reconstruction tasks in LLMs, where you mask one of the tokens and you try to reproduce that token, or you try to pick the next token. Same thing applies in vision transformers. The same thing applies in DNA sequence models and in transcriptomic models or MRI models. So what do you do then? Okay, now you've trained this and it's sort of the pre-training phase of the foundation model. Well, then what you do is you go tap one of the layers of that model and you visualize the embedding space. So here are 3 examples from papers I've written. The first one is looking at gene expression, RNA-seq, as I described in the beginning, which is gene expression using this Tahoe X1 model, which is not a model I made. It's a public model by this company, Tahoe. And what you see is that in the embedding space, the points, each data point, which is an entire gene expression vector, tends to cluster by cell type. And so there's very detailed cell types, and you can go multiple levels of the hierarchy to figure out how accurately it's representing cell type. Similarly, there's another model, Virchow2, which is made by MSR and Microsoft Research, and each dot here represents a patch of a histopathology image, a tissue microscopy image. And you can see that I'm sorry, the legend is a little low, but it's from a scale of blue to, let's say, a hot pink is Gleason grade, which is a cancer grading scale. And you can see that, okay, there are clusters of high cancer grade and low cancer grade and so on. So maybe there's some structure in the model. The last one is a model that I made, which is, of course, maybe like 5 years ago now, which is not great, but it's a much more difficult problem. Is trying to predict that same score, but looking at it from MRI using, hopefully a big model and a large data set. And it works in some cases. And of course, you can visualize it, but we can really go and benchmark it and evaluate it. And the way that's done today is by applying task heads to probe the accuracy of the model in essentially reconstructing or being able to predict these tasks. Cell type, let's say, or cancer grade, as an example. And so depending on the benchmark you use and how difficult that task is expected to be, you could argue about how good and bad it is. One thing was really interesting, this model that we came out with, it's on arXiv, but we published this, we put this up there in September. It's called Pulsar, looking at grabbing gene expression from multiple cells out of blood, PBMCs. And what we're able to predict in this red bar is that we're able to predict your flu vaccine response better than any of the other models. So if I was giving a machine learning talk in general, or bio machine learning talk, this is where I could end the talk. I could tell you how awesome my model is, how well I trained it, why the architecture is important, and then I'd be done, right? We say, oh, we learned biology. I can predict your immune response. And that's true. I could tell you whether or not how well it's going to impact it. I could tell you how well the model is scaling as I put more data into it and I make these architectural choices. But in my opinion, this overly simplifies and massively underutilizes what deep learning models can do. In particular, where is the biology? What did we learn from those prediction tasks? What does that tell us about how things work together? As an example, if you couldn't predict the task or couldn't perform the benchmark, why not? Or which layer should I tap? And how do I use the knowledge that's supposedly hidden within the model to do something actionable in biology, like make a new therapy, let's say? So I guess my idea is that we should go back and look at the theory that we kind of summarized in the first part and try to see that can we use that to extract out knowledge from the network. And so this is kind of the current direction the last couple of years that I've been focused on. And basically the idea is exactly what I said in the beginning is you can start with a pre-trained model that we believe is good. It can predict all these things. And we're going to ask, what does it learn on its nodal functions? And the way we do that is taking large amounts of data. So it's really important to have an atlas or great coverage of the phenotypic space or have lots of high dimensional vectors and flow them into the network. And then we're going to go and probe inside the network to look at what the nodal functions are really looking at. And then importantly, we can compute these metrics like smoothness or density, which are two generalization metrics that we looked at before. And I'll hint at one other metric or one other thing that we can do with this. But basically, the idea is that by clustering the data or training density models on these nodal representations, we can learn where the data lies, where the data is dense, and how smooth the function is with respect to the output. So as an example, when we saw the embedding space of these models, and let me go back for a second, we saw the embedding space of the models. The fact that they cluster tells you that you can draw like a pretty simple line or boundary to separate the various classes, right? Which means it's smooth. It's not like a really complicated function, which would be an example of not being smooth. Okay, so the question is, how do you do this practically for models that we care about? Well, the first step is to get the DAG of the actual function. And it turns out this is actually a little bit harder, but there's some packages now in the last couple of years that have made this a lot easier. So here's a really simple example. You can write some Python code with a torch model. You can pass it into this function, and you can see every operation that happens. So given the input, you can get the add, the ReLU, the linear layer, and so on. It's kind of the graph, the computational graph, if you will, of that model. You can do the same thing for CNNs. You might be used to this cartoon of how convolutional neural network works, or a block diagram that might be in a paper. But here's what AlexNet actually looks like when you break it down to individual components. And I'm going to talk about this in just a second, but this is highly linearized and things are stacked together. How do you get like this individual kernel representation? Well, we need to do a little bit more specialized processing. And I'll mention that in just a second. But you can do the same thing for transformers. This is the cartoon of how a transformer works in the block that you might read in a paper. But this is the actual architecture. And what's pretty interesting is, here, I'll do it online for people online, but you can kind of see the K, Q, V branches of the transformer model in all the computations that it's doing. And again, we need special processing because these are really high-level matrix multiplications. You're not getting at the token level or really what is the basic unit that the model is processing. And of course, you can do this for a real model. So here's a universal cell embedding, which I think was just accepted yesterday after several years. But basically, it's basically the same components that I showed you, the block of the transformer, and you stack them multiple times, and you can see them kind of like at a high level. There's 5 layers or so, and then there's an output layer that is able to predict the final embedding, and if you want like a classification from it. But I told you in the beginning that you need low integrity for these nodes for this to work, to avoid the curse of dimensionality. So how do you do that? And importantly, we needed not only low integrity, but we wanted boundary control on the derivatives of each of the nodes that are inside the network. So the first thing I realized is that in order to do this analysis, you need to reduce analysis to the size of what I call the local receptive field, which is really just to say, to be able to look at the individual tokens or the inputs and outputs of every node. So as an example, in a CNN, when you're sliding that convolutional kernel around, it's really the same nodal function that's being applied multiple times. So applying this to the model, basically what you get, which is kind of interesting, is rather than giving an image, you get a single data point as the embedding, representing the embedding, you now get a point cloud. And this is really important and maybe hints at why convolutional networks and transformers are so effective. It's because you get this massive boost in the number of data points that are used to train these nodal functions. Same nodal function applying now on thousands of data points. And so, of course, you can color them. But importantly, because there's more structure in the space and you have this data rate boost, you can train local density models that work really well. So here's a really simple example. If you ignored everything I just told you about reducing to the size of the local receptive field and you try to cluster the embeddings from something really simple like a ResNet, you might say like, oh, the embedding space is really like not structured. Like I here ran like PCA on it. And it really doesn't look structured. All the classes are mixed up. That must be a really rough function. But if you apply this nodal analysis trick and you look at all the individual patches and you clustered those patches using these density models and you kept the homogeneous clusters with respect to each class, what you see is that actually the model separates those classes really, really well. And so it's actually key to being able to understand what the model is doing is to operate on the level, the size of the token that the model is actually looking at. It's not a high dimensional input to a transformer. It's individual tokens. For most of the operations, some of the operations are matrix multiplies. But importantly, in this low dimensional space where we have good data density and we can kind of understand things, we can define things better like dimension and SVM dimension or smoothness and so on. And so one example of that is, and actually I talked about this last year, but so I don't want to talk about it too much, but basically the idea is that you can define those notions of smoothness and data density using clustering and let's say an approximation of the Sobolev norm or the smoothness of each individual node. And what I showed basically is that you can get this broad correlation with how well the model is actually performing. Without ever looking at the final output values of the model, you can predict how well it's doing. So each line here is a different model that's trained differently or trained independently, and I can rank the models just based on some of these metrics. Not only can I rank the whole model over the performance on the whole data set, I can look at sample by sample and I can tell you if the model is going to accurately classify that sample or accurately predict that sample or not, just by looking at whether it's in bounds or out of bounds of the existing data density that I have. How far away is it? It works most of the time. Of course, you can be far away and that network happens to get it right, just like a polynomial that's wildly oscillating will sometimes get the right answer. But in general, it works really well, especially with this very small amount of tuning. So the last part of that is, what do we learn from that? Well, just like what we saw in transformers and a lot of this other literature, we can go back and look at the attention maps or the heat maps that the network is paying attention to and say, oh, this is the reason why it works. And so here's an example. Given an input to airplane, and rather than using something like GradCam, which is a popular approach for feature attribution, just by looking at the distance from each patch, each token to the closest cluster center, I can come up with like a really good outline. And so just, provide some intuition or confidence that, yeah, this is maybe really how the network is working, even high dimensions, by doing this trick of projecting to a lower dimensional space, projecting all the nodes to a lower dimensional space and training density models. This is good. But, this is very qualitative, right? How do I become more quantitative about it? And importantly, can I use that to learn something a little bit more about biology? So what I wanted to talk about in the next part is kind of, I don't know, I would say the latest work, right? Which is a little bit of work in progress, but I think there's some deep insights here. And basically the idea is I'm going to zoom out and think about how a network is working in general. And so the idea is that we have the DAG of the actual model and we can flow data through it. But as we saw in the low dimensional representations that are specific to individual classes, as example, you can cluster based on, we looked at the ResNet classes, but you can cluster on any number of classes like Gleason grade and so on. At any given time, only a portion of the representation space is being utilized. And so I can kind of enumerate or precipitate this idea by, instead of representing each node in the network as like a physical computation node in the computational graph, instead I'm going to represent each node as one of those cluster IDs, one of those representations that is at that node. just to figure out which part of the representation space is being accessed. And so what I find, what emerges from this, is as I flow data through the network, different samples will take different paths. They'll hit different parts of the representation space at every layer. And so I realized that actually, if you take the collection of all the traces, or I call these traces or all these paths, it collectively collectively represents how a network is encoding the entire input data set, or any part of the data set will have this unique path. So I'll give you like a really simple example. Taking ResNet, convolutional network, and I pick 5 layers, and I plot for a few samples what paths they take. In this case, at each layer, I cluster the representation into 4 groups. And so importantly, even though the points start at different areas of the input space, they all end up with the same output prediction. So the output prediction is degenerate. It doesn't capture the full diversity or granularity of what the input phenotype or the input vector is representing. So if you do this for all the data, you actually get this compilated graph that actually kind of looks like a neural network graph, right? And this is actually the intuition for like why you can represent, or why at least visually you can represent a network using its representation nodes as opposed to the physical nodes of the network. And so, okay, I did this with, really simple with just 4 groups. I can do it for 25 groups, and it's really complicated and it looks crazy. But if I separate them by class, what you can see is that each class has a set of traces that are associated with it that are unique to that class, that represent uniquely what the network is doing at each stage or each layer operation or block of the network. And of course, these are vector representations, so I can UMap them, and I can reason about how similar the traces are, and why they're similar or not, and which layers they're different in. But importantly, I can pull out some of the classes, and so here's an example where from the orange class, which happens to be horse, where 2 traces, they both end up in the horse prediction. But if I trace all the examples that correspond to this top path, and I trace all the examples that correspond to this bottom path, I find that they are phenotypically very clustered. For example, the first trace, all horses are looking right, and the second trace, all the horses are looking left. It's really surprising. This is how the network is encoding that information, right? And we think, oh, it's not biased because it's predicting horse really well. But the problem is the output class is degenerate, right? It doesn't know. So, for example, if you pick this, penultimate layer of the network and you said, I'm going to look at that embedding space, and you look at the clusters there and you pick the nearest neighbors, you'll get a bunch of horses. I'm pretty sure you'll get a bunch of horses. But they will be mixed up between horses looking right and horses looking left. But importantly, if I trace this path and I follow the path that the network is taking to make this decision, I can see that it's actually uniquely defining, or kind of like a type group of specifically which part of the high dimensional input space corresponds to horse. Okay, so this gives us a way to subtype the input space and reason about which parts of the high dimensional input space are valid, or how the network is representing them. So the idea that the question I'm going to ask is, like, does it work for other models? Specifically, does it work for transformers? Because we're all using those now. And does it work for biology? Like, what did I learn from, what can I do in biology using this concept? So the idea is an emphatic yes, you can do it. This is a vision transformer, and I've done the same thing, breaking down each nodal function into the token level nodal function, right? Each layer into a set of token level nodal functions, clustering those representations and tracing that representation throughout the network. And so I think this is like 5 layers or something. But you can find similarities. As an example, like, what are the similarities? I don't know. It's a little bit hard to say. I'm just picking 2 traces manually and visualizing them. But as an example, in this bird case, all the backgrounds are green, and in the bottom trace, all the backgrounds are white. So for some reason, the network decided to pay attention to that. What I think was really interesting, or another interesting one, is this boat case, or ship, where the top ones all have a superstructure in their boat, in the boat, and the bottom ones all have sails. So somehow the network is picking up on that and encoding that in this deep hierarchy. And what's really interesting about it is a lot of them, like especially this one, they only differ in one layer. That one layer is where it's being different. So maybe we could utilize this, right? So, okay, it works for vision transformers and natural images. Does it work for biology? So I ran this on universal cell embedding. I just mentioned a few slides ago. Basically, it's looking at gene expression data, and I used it on I ran cell by gene, which is a public repository of gene expression data that everyone contributes, which with 100 million examples. And I ran it through this network and it did this whole clustering, which is a big computational challenge. And it's a little bit hard to read, but every row here is a different cell type that's been annotated in that data set. So I'll pick a few that I thought were interesting. Oh, I guess I should say it's hard to know what the traces are representing here. I have to look at the metadata tags, and they're not, because unlike images, it's not visually interpretable, at least to me, because I'm not really a biologist. So you could look at the genes and then think about why the traces are different, but I could look at the metadata tags. So some of the ones I got a hit on are B cells and CD14 positive monocytes. And in both cases, the top trace corresponds to leukemia or has the annotation of leukemia, and the bottom trace has the label of normal. Similarly, for this cell type, the top trace, which is a little bit hard to see, so I'll highlight it here. They're really differing only in one layer, but this top bold trace and this bottom bold trace are differing in COVID-19 exposure. So the top traces have all been annotated. I think it was like 98% actually, to be honest. And the bottom trace is all normal. So it's a statistical correlation that I'm talking about, but it's still very interesting that I'm able to pull out these phenotypes, and the model was not trained on those outputs, just trained on this simple structural reconstruction task. So what do you do with this data? Well, the first thing that you could do is you could run differential gene expression between the different groups. So similarly to how we were looking at the different the traces, or visually looking at these pictures and reasoning about green background, white background, horses looking left and right. To do that quantitatively, we could look at something called differential gene expression, which basically looks at what are the genes that are different between these two subgroups and reason about which ones are being active and activated and why, and maybe the environment plays a role. And then the second part is when if we want to develop a therapy as an example, or really understand the causal factors that control that expression, we would want to use like CRISPR as an example. And do gene editing to figure out, can I move a cell that's in one state on one trace to a different trace? And so, just to give you a hint of what's possible, maybe in the first part, looking at differential gene expression, I mentioned this project where we're looking at prostate cancer with spatial transcriptomics and histopathology. And here's an example where this grad student from UCSF, Maggie, was able to find this signature by correlating histopathology with spatial transcriptomics, and specifically a gene signature, which they call interferon gamma signaling. And so what I was able to show is the patch embeddings or the trace embeddings from like an existing histopathology model. And it's a little bit hard to see here, but I can do it with IFN gamma and this other signaling pathway called TFN alpha. And it's a little bit hard to see here, but basically I'm able to automatically annotate regions of that trace UMAP that correspond to high signals. And so importantly, it's really a small portion. So each one of these pictures here is a biopsy slide. So a prostate biopsy, you get a piece of tissue, you put it on a slide, you're able to take an image of it, and you're able to run spatial transcriptomics. And what I'm saying here is that just from looking at the histopathology, I'm able to predict these gene expression scores because I'm able to subtype or very finely subtype the output of the prediction. So the question that we have, and this is kind of ongoing work, is how does the presence of this signal or the fact that you're in these different subtypes affect a patient's response to therapy? So it could be chemotherapy, radiotherapy, immunotherapy. And importantly, how does that affect their long-term survivorship, whether their cancer relapses? And if you could predict that, then for an individual patient, once you do this sort of subtype, and not only knowing what the model says the final prediction is, but by subtyping their disease and saying you have the specific type of the disease that we know or tends to correlate with good therapy response, then you can perhaps pick the best therapy. So this is still retrospective, right? And it's very correlational. And so the other thing that I pointed to was, can we cue causal experiments? Can we do causal experiments? And this is a collaboration that I have with Richard James, that's at Seattle Children's, and Dina DiCarlo at UCLA, and they actually developed the first part of this pipeline. And the first part of the pipeline is using nanovials to measure individual secretion specifically IgN, which is an antibody in B cells. And so they have a platform to do that. And Rich deals with culturing all these cells and being able to measure them. And the idea is that actually Rich has this therapy where they're able to grow in vitro. These cells are optimized their secretion, the antibody secretion, and they can inject them into the bone marrow of mice, but also humans. And they've done it in humans, and it really works to give them immunotherapy resistance. So they're, especially in people that have their immune systems compromised due to drugs or cancer or whatever, using this therapy, they can have an active immune system for, I think they showed for up to like a year or two. So the question is, how do we optimize that, make those cells secrete longer and so on? And so basically what I started looking at is, can we look at traces and can we understand what the differences are in secretion level? The problem was kind of with their method is they're not able to optimize it. Every time you grow a culture of cells, some of them will secrete a lot, some of them will secrete little. So what we asked is there a genetic basis for why secretion occurs, and can we cue causal experiments? So we have sort of the basis of this. We can look at the embeddings with a bunch of foundation models. We can extract traces that correspond to high secretors and low secretors. We can identify the genes that are specific or hypothesize or that are different between those two different groups. And then we can, right now what we're trying to do is cue these experiments where we're actually editing those genes to see if they're actually controlling secretion. So this is ongoing work as well. The last thing I'd talk about is, you might have heard about this. This is something cool that we put up on Archive last year, but where we're looking at DNA sequence models. And actually, the literature is a little bit more robust here, where what folks are doing is they're training these DNA sequence to function models. And what that really means is given it literally ATGC of your DNA sequence or DNA sequence reference genome of a species, trying to predict the activity in every cell type. And what I mean by activity, I mean gene expression, or it could be anything, but it could be gene expression or or chromatin accessibility specifically. But based on that model, then they do this sort of introspection step where they try to attribute different parts of the input to what's causing peaks or spikes in the output predicted signal. And using that, they can essentially, and this is kind of what this image from their paper is showing, but for different gene expression data, you can highlight which parts of the DNA sequence were thought to influence the expression of that gene or not. So they call those enhancers. But what's really cool about that is you can find those enhancers, then you can literally synthesize DNA and find a way to deliver that into patients. And they use viral vectors, like viruses, essentially, to inject them into like a mouse. And you can go and visualize and ask, did it really express the gene the way I thought it would? And this really gets at causality, right? Like, I thought I made this prediction based on this model that this part of the DNA sequence is going to influence this, is going to increase the expression or decrease the expression of this gene. And they go and measure it, and they literally make an image and say, oh yeah, this cell type, I know it's the cell type in this location of your brain or your body or whatever. And you can visualize whether or not it worked. And so this is still ongoing work, and there's a whole institute now. You might have heard of C-Hub, which is a collaboration between Chan Zuckerberg, Allen Institute, and UW, and a lot of UW faculty there as well that are actually focused on this and kind of expanding this into new domains. So kind of my last slide is, where do we go from here? Like I told you a lot of things. What do we do? Well, I showed you some positive results from nodal analysis of these networks that you can extract out these traces. You can predict the generalizability, and then hopefully that cues experiments. But what is the work that remains to be done? Well, the first thing is that I kind of cherry picked which layers to look at, because a deep network is really, really large. There's sometimes millions of nodes, but I think it's order of hundreds of thousands. And depending on the type of data that you flow through it, actually even just learning the density model on each nodal function is super expensive. And so, okay, I kind of abstracted a lot of that and masked over what those challenges are and how do you climb over them. But kind of stepping back a little bit, I think the question that we need to answer is, which layers should we be looking at and what level of abstraction should we be applying? So as an example, maybe in your network you have the addition operation. I took a vector and I added 5 to it. Should I really be looking at that nodal function? Is that important? Or is it just like a simple shift and I should skip over it? That type of logic haven't implemented, and we need to figure out, how do we think about this? What's the right level of abstraction? Is it a whole layer? I should look at the embeddings of every layer, or is it a block, like a convolutional kernel, or just, as an example, just the transformer block? Or is it really just you have to do every operation? I don't know the answer to that, but it is very expensive. So being able to boil that down to the right level of abstraction is kind of the key. The second thing that I hadn't really talked about, I'm able to draw these traces and follow the paths, but I don't really know what the paths are doing. Why is it separating horses looking right from horses looking left? Why is that a useful thing for the network to do? Oh, you could say, well, it's minimizing the loss function, so that's the reason. And sure, that maybe there's dynamics of training dynamics that are associated with that in SGD, and we can go down the optimization route. But what it doesn't tell us is why that's a useful computation, right? So as an example, maybe that's implementing an OR operation or something a little bit more complicated. Importantly, if I'm able to break down models into small primitives on these representation spaces, you can look at compositional logic and maybe define, let's say, a language of what the model is literally looking at. And what are the propositional logic? Like if I have a token here and if I have a token here, then it means I get this part of the output space gets hit. That sort of logic, I think, would really solidify. And so importantly, if we understand that, then we can start looking at how that maps to real biological circuits that we know about. Like this gene interacts with this gene, and it causes this gene to be expressed, as an example. And that's literally what we were looking at in the transcriptomic UCE model case. Third part, I think, is probably the most important, is that I've done this right now on observational data. 100 million cells is something I mentioned, but it's not interventional data. So how can I make interventional choices based on observational data? This is the whole realm of causal inference, and it opens up a big can of worms that I don't know if I'm quite ready to get into. But the thing that I can say is that, okay, well, maybe we can do perturbation and then we can go test it in the wet lab. There's obviously a gap there of how quickly I can come up with hypotheses of what the different subgroups are and what genes to perturb, and how quickly I can actually test those in the lab to validate that those hypotheses were right. There's a big gap between those things, and maybe robotics will improve and we can do that much quicker, but it's not quite there yet. I guess one way that I would mention, and especially if you're interested in this area of in silico perturbation, is there's this, I don't want to call it a field, but this idea called mechanistic interpretability that was coined by Chris Olah, who's now at Anthropic. But the idea is that they're also looking at what's the causality or computational circuits that are within models. But the way they're doing that is it's really been focused mostly in the LLM space, but they're looking at if I change one token within the network, how does it cause the final output of the network to change? So as an example, you type a prompt into ChatGPT and you say the sky is, I don't know, you can fill in the blank. Maybe you say blue if you're in Los Angeles. Maybe you say it's gray because we're in Seattle. But what they're looking at is how does one neuron, or they call it neurons, but it's really just a token within the network, how does perturbing a token or perturbing one of those nodal functions affect the final answer to cause it to say, oh, the sky is green, or, the capital of Paris is Texas, is Austin or something. So that's the type of examples that they're looking at, is how I think it is largely related. I think the difference between what we're doing is we're using observational data to learn those kind of statistical relationships directly, and hoping that, and I can show, for example, if I push a token that's in one part of the input space directly into another part of the input space, I know exactly what it'll do because I have a prior example that followed the exact same trace. It's deterministic. But if I push it to a part of the input space that I haven't seen before, I don't have any examples of it, then what? And that's kind of what their mechanistic interpretability work is looking at. But there's a bridge here between those two approaches, especially when you go and test it in the real world. And the final thing that, I guess I hinted at is really what we're after is being able to design personalized medicines, right? And so when we do that and we train these big models, what we ultimately want to ask is, when you go measure your data and you go to UW and hope you get a blood draw and you get gene expression data or you get protein counts or whatever, how relevant is your data? Which trace do you correspond to on that graph? And so to answer that question, we need a lot more data, but also we need a good way to annotate that data to be able to understand whether that sort of association is worthwhile. So with that, I just wanted to thank my collaborators and my funding sources, and I'm happy to take any questions. Hey.
If you give some input, all the nodes that were somewhat, so you're just imagining that like what's the most active.
Yeah, so that's a really good point. So that's why I changed the representation in the graph. Instead of looking at which nodes get activated, I look at which representations get activated. So this distinction there, okay, yeah, that's a good question. All right, so let's say this is like a neural network diagram, right? Like that's what you're used to, where the block is a matrix multiply. So like this high dimensional there's whatever vector, you do a matrix multiply, it actually touches all of the nodes, right? But what I'm doing is taking the output of all of these nodes, and then I'm projecting it to a lower dimensional space, training density models in there, and saying, okay, yeah, it does touch all of the nodes, but those nodes have a dual in this representation space. So for example, you put in a vector and you get all ones. You put in a different vector, you get all zeros. Those are very distinct. And so maybe all ones you map to like node A1, and all zeros you map to A4. And like similarly, you essentially segment the representation space. And the reason that's reasonable to do is I can actually recast this transform as a series of transforms using a frame as opposed to like a basis. And I can project the input vector into specifically this. Does that make sense? Okay. I wish I had a, yeah. I'm making a dual network. Yeah. That has it looks the same, except instead of physically which node, like physically those nodes.
Yes. There's a lot of activation.
Correct.
A lot of things. And then you're collapsing that in some other.
Into a low dimensional space. And you have to train that other thing. Absolutely. And I think that's what I was trying to say, here. Yeah, basically we're tapping those representations and training this model. And the reason why, and I guess what I added just now is the reason why it's reasonable to do that is because I could convert every node, every layer into a series of smaller layers that project specifically to those parts of the representation. I can segment the representation space. And that's literally what the density models are telling us. Yes. Exactly. In fact, when I do that, I can preserve the exact to epsilon accuracy, the exact operation of the network. So I can convert the network to like this dual network that's actually made up of these smaller nodal functions that's a trick sleight of hand that I'm doing. But yes, absolutely. Okay. Did I answer your question? Yes. Yeah. So each item here in this thing is like a part of the representation space at that layer that's being activated. So when we look at these traces, it's like it really is that part of the representation space. And it's not all of them. It's a really specific small portion where we have data density. Yeah, and it's the same layers or the same nodes, but yeah, right. The y-axis basically has been remapped in some sense. Yeah, exactly. Hey.
So, and applied to this, you showed some like TC or UMAP.
Yes.
So now is the y-axis your like TC or UMAP clusters?
Exactly. It's exactly what it is. The best part about this is that there's no magic in it. You can try it at home. I could do it tomorrow and it'll work. And I think the insight is that you just need to do it at every layer. And I don't know why that was been something that people missed before. They only looked at one embeddings. Like, why look at the class? Yeah, why look at the output layer? Why look at the class token? How did they come up with that? They're not even, in some cases, they're not even training that part. So as an example, I showed like the UMAP, I showed this MRI plot with dots and they're kind of overlapped. I picked the middle layer of the network, like an autoencoder, pick the middle layer. Why do you pick the middle layer? Who came up with that? Oh, they say, okay, it's bottleneck layer. That's where the information gets compressed. Okay, but then it should exist at the next layer too, right? It's either masking the information or it's retaining the information, right? And so this is just a way to like surface that all out at every layer. And importantly, I do think that the networks are filtering information, right? The output is degenerate. Like these things all end up as bird. But by following the path, we can identify, okay, actually, it's a little bit more nuanced than that. And the idea is that it's like a funnel, right? There's a really big high-dimensional input space. And it's actually only using certain portions of it because that's where you had data samples. Or importantly, at the first layer, it projects it down to a representation in which the representation space can actually be clustered, and you can define density models. And then it kind of goes down this funnel until it gets to the final degenerate output, which is bird or whatever the prediction of the network might be.
Question?
I think there's a question online.
Yeah, just wondering, this seems to be data dependent. If for one input data set, you may have, like the red lines show, a big divergence, which shows, some unique correlation but
Yeah.
for a different input set of data, you may have a totally different set of lines.
Totally. That's a great point. And I bring this
So
up
how
at
do
the
you
end.
how do you make a conclusion? Yeah, if it's
Yeah, no, that's a great question. Right. So the maybe pitfall of this method is that it's absolutely data defined. You have to use the data together with the weights to analyze the network. The network but I mean, I guess the insight here is that you can't just analyze the weights of the network. It doesn't tell you enough. It doesn't tell you which parts of the representation space are being hit because there's no way to quantify what the input space is. The input space is just really large. We have no bounds on which part of like Rn we're occupying. So I guess that's my pushback, but you're absolutely right that it is data dependent. And so my solution to that so far has been to collect really big data sets and push it through. But importantly, by doing this iteratively, you can actually represent a large data set essentially using like prototypes or what are like the canonical examples that you need to represent all the unique traces in the network. So maybe you need one from each of the traces as an example. And if you retrain a model using that, do you achieve the same performance? I didn't show this result, but actually we do have some results on that looking at online training where you use the traces and then you can queue which round of experiments to do next, like what data to collect. So basically some of these traces will have more data associated with them than others. So you could use that as a scheme, but
pattern.
Yeah, but yeah, I think it was a great point. I don't have a good answer, but maybe there's some work to be done there in analyzing what the circuits are, and maybe then it becomes not data dependent.
Thank you.
Yep.
Hi. Sorry.
Showing very complicated loss curves, and then you add Sobolev norm, and suddenly they're convex-ish.
Okay.
How does that work?
Okay. Yeah, so this example is basically I only give input-output pairs. I take the DAG of the true function, which is max, which is basically two inputs come in and you have like a neural network, like a matrix multiply, let's say, or specifically I use Chebyshev polynomials. So it can do crazier things, right? And you try to train it, and you try to find the parameters of this polynomial that fits the input-output pairs. And so in this first example, which is just two inputs, two-dimensional problem, right? Just if you limit the amount of input-output pairs you get, and you look at, and then you visualize the polynomials over the whole grid of the 2D, you see that actually the function, the polynomial that it learned is like not stable. It learned some wild oscillations because there's no control on the derivative or the smoothness. It knows nothing about the smoothness class of the target function you're trying to approximate. And so you can do the same thing. And so, but then what I showed is that if you specifically try to minimize, in addition to matching the training data points, the values, you also try to minimize the Sobolev norm, which is essentially the gradient of that node, then you get much better control and it converges really well. And importantly, if you actually use real optimizer instead of like stochastic gradient descent, and use convex optimization or whatever, you can guarantee that you match the data points and you'll do much better because it'll try to minimize the Sobolev norm after meeting the epsilon machine precision or whatever the data consistency threshold that you set, right? If you use convex optimization stuff. So the same thing applies in deep networks, where at each node, so you given 4 inputs and one output, what's the max of these 4 inputs, and you try to train a polynomial to do this complicated thing, but you're given limited data. You can't densely sample the whole grid, or even if you do, you can't do it fine enough. So if you check the points in between where you had data points, you see actually if you sample those, you see that you have wild oscillations in function that it ran. And in this particular case, I know what the real function is. I know that it should be like you can really represent max 4 or max 8 or max whatever using a bunch of two-dimensional maxes. And so that is what this shows, is that I didn't actually tell it that, each one should be max. I just trained it and just controlled the derivative by minimizing the Sobolev norm. And what I find is that, yeah, it basically learns max. It learns a network of these max 2s. But importantly, it's not just like visual. You can look at the error and you can compute the error, and it seems to work much, much better. I think I have other examples, but this is the example where it's like it's very visualizable. Right? You can see it's two-dimensional. Each node is 2D. Yeah, does that make sense? Okay. Happy to talk about it after. Yeah.
Hello?
Yes, hello.
Hi. What paper is this presentation over? Like, did you write a paper on this?
There's several. Yeah. So this part actually is in my thesis. And there's a couple papers on archive and presented in workshops and all that. Some of these other things are in papers that I've published, which are like, let's say, applied part of it. And this part is in a manuscript that should show up on archive. Okay, this part is in a paper at middle. And this part is in the manuscript that I'm about to submit in a week or two.
Okay.
So
What about
you'll
slide
see it.
17?
Yes, it'll be in the manuscript, and I'll happy to send it to you.
Thank you. I would like that.
Yeah. I will send. Okay. Perfect. I will do that. Let's do that. Okay. Yeah. Feel free to email me. Hopefully you have my email. I'm happy to talk after or.
Questions, let us know. We're gonna get into the schedule.